All right, hello everyone, and welcome back to lectures 2 and 3 for week 11, chapter 14. Let's go ahead and get into it. So, we will be talking in lecture 2 about the dissociating memory circuits. Carl Lashley, he searched unsuccessfully for the neural circuits underlying memory, found that the severity of memory disturbance related to size uh, instead, rather than location of the injury. Then William Scoville, he performed a bilateral medial temporal resection of a young man, Henry Molaison. HM is the initials that are usually used to refer to this patient. And this was due to seizures that had originated in the region of the amygdala, hippocampal formation, and the associated subcortical structures. So Scoville removed them bilaterally. Now, after the surgery, HM had severe amnesia, lacking explicit memory. So he did not recall anything that had happened after the surgery. Now, despite this deficit, HM had actually an above average IQ. He performed well on perceptual tests and could recall events from his childhood. HM's performance on implicit memory tests was left intact. Now, HM's right hemisphere lesion is highlighted in the brain view ventrally. We're looking over here, and the lesion runs across the wall of the medial temporal lobe. On the left side of the brain, has been shown to be left intact to show the relative location of the medial temporal structures. Parts A, B, and C are based on MRI scans, which depict a series of the coronal sections of HM's brain. Another interesting patient is patient, and this is one where they definitely have some documentaries on HM as well. It's really interesting to watch him because he's not able to make new memories. He will be going over and repeating things that he had just said before. Every single day is truly it's a new awakening into the life that he has around him, and you can imagine how frustrating that would be for people around him too, if each day you wake up and you're in a new place or with new people who you don't recognize, and how unsettling that may be or that may feel. But he was a great musician. He was a fantastic pianist, and he could still today, he may say, I don't know how to play the piano, but they would put a piano in front of him and ask him to play, and he could just play beautifully. So I would encourage you to check that out if you have a chance. Additionally, we have patient JK. This patient had impaired implicit memory with intact explicit memory. So this patient had developed Parkinson's disease in his mid-70s and started to have memory problems at age 78. He had damage to the basal ganglia, and he showed an impaired ability to perform tasks that he had done all of his life, like just turning off the radio, something that may seem as easy and as simple as that. He could no longer do, but he could recall explicit events. So the intact long-term memory displayed by JK was really vastly different from HM's situation. While HM would not have remembered anyone who was coming even 5 minutes after being told of their expected arrival, instead, because of the Parkinson's disease, which primarily affects the basal ganglia, JK's deficit in implicit memory was probably related then to his basal ganglia dysfunction and had to do with more of those procedural type tasks. There's also this patient with Boswell's amnesia. Boswell, he developed herpes simplex encephalitis, had a recovery from the acute symptoms, had low average intelligence from neurological infection, and severe amnestic syndrome in that case. So he completed 13 years of schooling. He had worked for nearly 30 years in newspaper advertising. He was a normal, well-adjusted person. He was successful in his profession by all accounts, but then after his infection, his speech and language remained typical, and he showed no perceptual movement deficits. Now, Boswell's sensory and motor cortices are intact, as are his basal ganglia, but his injuries are more extensive than HM's. Like HM, he has no new memories, but unlike HM, he has severe loss of access to old memories. Since recovering from that uh, herpes simplex encephalitis infection, patient Boswell has had great difficulty remembering events before and after his illness. Areas of damage in the medial temporal region, basal forebrain, and posterior orbitofrontal cortex are highlighted in red over here, which you're able to see some of those areas of damage. And these are really some key regions for some of that memory functioning. And like we said, the amnesia and the brain pathology that was documented by Antonio Damasio and colleagues, it showed that critical damage of bilateral destruction of the temporal medial regions, the loss of the basal forebrain and posterior part of the orbitofrontal cortex, and insular cortex from the lateral fissure. All right. This again just shows a view of his brain on imaging. And we will go ahead and move on to part 3, lecture 3. So in this lecture, we'll be going over the neural circuit for explicit memories. These are the primary structures for explicit memory are all listed here. So the medial temporal structures of participating in memory in monkeys. In figure A, we have this ventral view of a rhesus monkey brain. On the left side, the medial temporal regions were shown. Each of these regions plays a distinct role in processing sensory information for memory storage. And on the right side, there is the hippocampus and the amygdala, which are not directly visible from the brain's surface. Instead, they lie within the cortical regions that are illustrated on the left. So in these two areas, it's really if we're zooming into this section a little bit further, or we're cutting some of this away, we're able to expose the hippocampus and the amygdala there. All the structures are present on both the left and right sides of the brain. In this figure B, we can see this flow of information from the sensory cortices, first to the parahippocampal and perineal regions, then to the entorhinal cortex, and finally to the hippocampus. The hippocampus feeds back the medial temporal regions, and then to the neocortical sensory regions. We're going to take some of those specific brain regions now bit by bit. Let's start with the parahippocampal cortex. So this is the area of the brain that receives connections from the parietal cortex. It's believed to take part in visual spatial processing. The pararhinal cortex receives connections from the visual regions of the ventral stream. It's believed to take part in visual object memory. The entorhinal cortex receives projections from the parahippocampal and perineal cortices. It has this integrative function that's the first area to show some of the cell death in Alzheimer's disease, notably. When we're talking about the visual spatial memory, this is using visual information to identify an object's location in space. So lab animals and human patients with selective hippocampal injury, they typically show this severe deficit in various forms of spatial memory. For example, monkeys with parahippocampal lesions have difficulty learning the locations of objects. Animals with especially good spatial memory, they should have bigger hippocampi than species with poorer spatial memory. Monkeys with, like I said, that parahippocampal lesions have difficulty learning the locations of objects. This is typically in like a visual recognition type task, or where there's object that's placed in a certain position and the monkey has to recognize that. The hippocampal formation in food storing birds and rodents is larger than that of birds who are not food catchers and rodents who do not store food. And again, this makes sense because in these cases, these birds and rodents, they need to place that food somewhere and they need to make sure they're able to get back to that specific area to get the food at a later date. So if you don't have that, it's going to be trouble for you. And there's also been this really, really interesting study you may have heard of before where these London taxi drivers, they were examined because the amount of work it takes to become a taxi driver in London is incredibly extensive. It requires really memorizing all of the streets and crossroads throughout London. And upon MRI, researchers found the posterior region of the hippocampus in these individuals is significantly larger than the same region in control participants. Now, does this mean that people with larger posterior hippocampi are going to tend to be taxi drivers in London? No. Remember, we're talking about the experiences that an individual has and the impact that can have on the brain structurally. And that's that plasticity, right? The neuroplasticity. So instead, these individuals have spent so much time and they've needed to hone their spatial reasoning skills. They've led to this actual structural change in their brain to reflect that experience. So we talked a little bit about that object memory type task. This is where a monkey is shown an object in front of them. Then it displaces the object to obtain a food reward. Then a monkey is trained to displace an object to get the food reward. It's then shown two objects. The task is to displace the new object to obtain the reward in like the visual recognition task. The monkey is then shown one object to displace for a food reward in an object position task. And on the next trial, the monkey is shown two identical objects, and they must choose the one at the same location as the initial presentation. So in all of these, it's trying to understand the learning that's taking place, where we're saying, is the monkey able to learn what is the new object, what is the old object, or is the monkey able to identify, well, what is the location of the object that has food underneath it versus the one that doesn't? This graph, it just goes back to that discussion we had about the food storing versus non-food storing birds in the hippocampal volume of the forebrain in these two types of families of songbirds. Birds that end up storing their food, like the black-capped chickadee, have hippocampi that are about twice as large as birds that are not catchers, like the sparrow. For the different spatial cells in hippocampal formation, there's really three classes in the rat and mouse hippocampus, medial and entorhinal cortices that have been identified. There are place cells, which discharge when a rat are in a spatial location, regardless of orientation. There's the head direction cells, which discharge whenever a rat's head points in a particular direction. And there are grid cells, which discharge at many locations, forming a virtual grid invariant to changes in the rat's direction, movement, or speed. So the place cells and head direction cells are found in the hippocampus, and they're closely related structures. The grid cells are found in the entorhinal cortex, major afferent route into the hippocampus. And as we're looking at this figure here, at the right in each part, the XY coordinates indicate the directional selectivity of the cell recorded at the left, which is shown A and B. The place cells discharge when a rat is at a spatial location, regardless of its orientation, which we're able to see. With C, the head direction cells discharge when a rat's head points in a given direction, irrespective of its location. And with D, the grid cells discharge at many locations, forming a virtual grid invariant in the face of changes in the rat's direction, movement, or speed. The neocortex, it projects to the entorhinal cortex, which then projects back to the neocortex if we think back to that the little diagram. So the signals from the medial temporal regions of the cortical sensory areas keep the sensory experience alive in the brain, and the neural record outlasts the experience. The pathway back to the neocortex means that it kept apprised of the information process in the medial temporal regions. The Frontal lobe's role in explicit memory is subtler than that of the medial temporal lobe, which is helpful to note. The frontal lobe appears to participate in many forms of short-term memory. So all sensory systems project to the frontal lobes, and during tasks in which monkeys must keep information in short-term memory over a delay, certain cells in the frontal cortex will fire throughout that delay. So animals that have not learned the task end up showing no such cell activity. There's this interesting syndrome that exists called Korsakoff syndrome, and this is the permanent loss of the ability to learn new information. So it's this anterograde amnesia, and to retrieve old information, retrograde amnesia. And it's caused by diencephalic damage due to chronic alcoholism or malnutrition that produces a vitamin B1 deficiency. The memory disturbance is probably severe because the damage includes areas of the forebrain and the brainstem structures. Now, researcher Mortimer Mishkin and his colleagues found that the neural circuit for explicit memory incorporates evidence from both humans and laboratory animals with injuries to the temporal and frontal lobes. And here's a graph showing the general anatomical areas controlling explicit memory. And in figure B, that diagram shows the flow of information that begins on the right with the inputs from the sensory and motor systems, which are not considered part of the explicit memory circuit itself. And the relationship between then where that information comes in and those neuroanatomical regions. Sensory and motor neocortical areas connect to the medial temporal regions. The basal forebrain structures play a role in maintaining appropriate activity levels in other forebrain structures to process information. And the temporal lobe is hypothesized to be central to long-term explicit memory formation. The prefrontal cortex is central to maintaining temporary or short-term explicit memories and the memory of or for recency, which is kind of the chronological order of explicit events. As a bit of a review, some of the consolidation of explicit memories. So the hippocampus consolidates new memories, and the consolidation or stabilizing memory trace after learning, memories move from the hippocampus to diffuse regions of the neocortex. Once memories move, the hippocampal involvement is no longer needed. When memory is then replayed in your mind, it is open to further consolidation, which is called then reconsolidation. The new information is constantly being integrated into existing memory networks, and it is possible to erase negative memories using amnestic agents when the memory is revisited. For example, individuals with PTSD. I think that's an area where there is some ongoing research trying to understand, is this a process that we are able to use therapeutically? So this shows the proposed neural circuit for explicit memory, including the temporal lobe structures, the frontal lobe structures, the medial thalamus, the basal forebrain activating systems. The figure B that we have shows the circuit diagram showing the one-way flow of implicit information, beginning with the inputs from the sensory and motor systems that are not considered part of the memory circuit. The basal ganglia receive input from the entire neocortex and send projections first to the ventral thalamus and then to the premotor cortex. The basal ganglia also receive widely and densely distributed projections from the dopamine producing cells in the substantia nigra. Dopamine appears necessary for basal ganglia circuit to function and may indirectly participate in implicit memory formation. The amygdala is really a key structure in the emotional memory, and in figure B, there's this circuit diagram that's showing the information flow in the emotional memory, and we can see again the importance of the amygdala specifically for this type of memory. Now, there is this interesting unconscious nature of the implicit memories, and Mishkin believes that the implicit memories are unconscious because the connections between the basal ganglia and the cortex are unidirectional. So the basal ganglia receives information from the cortex, but it does not project back to the cortex. For memories to be conscious, there must be this feedback to the cortex, and the medial temporal lobe projects back to the cortex. So explicit memories are then conscious. With some of that emotional memory, there's the memory for the affective properties of stimuli or events. These can be implicit or explicit. And like I had said before, the amygdala is really critical for emotional memory. And damage to the amygdala then abolishes emotional memory, but it has little effect on implicit or explicit memory. That's partly because the amygdala has really close connections with the medial temporal cortical structures and the rest of the cortex. It sends projections to the brainstem structures that control autonomic responses such as blood pressure and heart rate, the thalamus, which controls hormonal systems, the periaqueductal gray matter, which affects pain perception, and the basal ganglia to tap into the implicit memory system. This is that unidirectional neural circuit proposed for implicit memory. We've seen this figure before, just a little bit earlier as well. So I don't know if it bears repeating. Emotionally arousing experiences tend to be remembered vividly. So emotionally driven neurochemical and hormonal activating systems, which are probably cholinergic and noradrenergic, then stimulate the amygdala. The amygdala, in turn, modulates the laying down of emotional memory circuits in the rest of the brain, especially in the medial temporal and prefrontal regions and in the basal ganglia. Now, there's been this evolution of the accretion model of memory that was proposed by these researchers up here, Murray, Weiss, and Graham. And they proposed a model for the organization of memory systems that differs dramatically from those described previously, where several memory systems arose during the evolution to exploit specific opportunities at particular times and places. And this type of exploitation has then led to radical changes in the cerebral anatomy and function, where virtually all cortical areas are involved in memory. This way of experiencing and encoding memory is true explicit memory, and it is unique to humans. And that's partly why there's believed to be this large expansion of the human brain, especially in those prefrontal regions and the related connections with the hippocampal regions, which not only encodes experiences, but embeds them in time and in our concept of self. So over the course of human evolution, it's believed that this significant change in cerebral anatomy resulted in the emergence of the many unique cognitive abilities that humans possess, again, including language. Now, that is the end of lecture 3. I'll see you later for lecture 4.